Your life. Okay. And okay, fine. Yeah, thank you. Yes. Hello, everyone. I hope that all greetings for the day. I hope everyone is fine because of the ongoing current corona situation. I guess that all the people are safe and they are fine. And I would say that please take care of yourself and your family and ensure that you take the basic what you call the precautions in order to avoid the corona situation. And we all sincerely pray and wish that all of you are safe and you can do your work properly and get yourself done with the things, keeping in a healthy mood and in a healthy aspect of life. Okay, let us start with the presentation. Today I'm going to talk about as how to write research methodology. What are the basic aspects which are associated with it and how we go about doing the whole process. So this webinar is a complete guide on how to write research methodology and how exactly can we frame it out so that it will be suitable and useful for writing and guiding people while evaluating the thesis. So the whole presentation here today that I have been organized in a systematic manner with the various topics and outlines being put forth. Now here is the content that is being presented today. They are the multiple aspects which are shown here. Now first we basically talk about the idea of research, because even before we propose for research methodology, it is important that we talk about what is research, why we need to do research, and what are the various aspects associated with the research that have to be done. So the basics for doing the research and how it needs to be done is definitively has to be mentioned first. So after talking about research and what it is and why it needs to be done, we talk about distinct types of research that we are going to do. In this, we will be covering about the various kinds of research that are done online as well as by people around the world, and how are they and what they are. Now, once we have an idea of different kinds of research which is being done, we now come with the outline of research. That is, what exactly is this, how exactly the research has to be carried out in a systematic fashion, how we need to proceed ahead and how we need to do, what are the various aspects which have to be considered and emphasized. And in this, one of the aspects comes out is the methodological approach. So the overall methodological approach for doing the research, how it needs to be done and how it needs to happen will be put forth. Once we explain this aspect pertaining to research methodology, then we come to the introduction to the concept of research methodology with its significance. Why one needs to have research methodology written, what are the various aspects that are associated with the research methodology, and what is the significance of this particular aspect. Now, as said here, RM, which is otherwise for research methodology, which has been short formed here, for the different kinds of research, you have different kinds of methodology which is outlined and which is written. So we are going to discuss each of the research as how to do the methodology part and how we go ahead and how we present it. Then research methodology is a very systematic pattern of writing, and it needs to be written in a particular manner so that everyone understands the basis and the various aspects related to it. So there are steps in research methodology which are involved, which include introduction, methods for analysis of data, then methods for concluding based on analysis of data, and logical interpretations based on the conclusion and whether they meet our expected outcome or not. So all these various aspects will be discussed in detail in today's presentation, and we will be covering each of them in detail in order to understand how this happens. Now, before we go to understand about research methodology, it is essential to understand what is research. Now, research, typically there is no specific single definition for research, or there's no single consensus for defining research. Multiple people have utilized multiple methods for explaining about research. Now, if you go and look at Merriam-Webster dictionary, the classical definition of research is like it is a systematic inquiry into a particular problem which involves investigating or experimentation. This ultimately leads to discovery and interpretation of facts. Now, so it's a studious inquiry or examination, which especially investigation or experimentation aimed at the discovery and interpretation of facts. And this in itself is completely sufficient enough to explain that it's a very systematic and methodological method. As per John W. Kresser, research is a process of steps used to collect and analyze information to increase our understanding of a topic or issue. Now, this is one of the best definitions which can be given for research. When we say research, it is a process of steps definitively, and any research consists of three distinct steps. One is to pose the research question. Two is to collect the data pertaining to the question, and three is to answer the question. So, uh, posing the question, collecting the data, posing the question, collecting the data, and answering the question are the three important aspects which are associated with the research. And these are the ones which have to be used typically in order for understanding the whole process. Okay. Um, okay, because of the voice issues, I'm again starting back from the first part as what is research from the previous slide. Now, the operational definition of research, there is no single consensus which is associated with it. They are multiple ways by which people have defined the research, multiple ways by which people have defined research. And as per the classic definition by Merriam-Webster Dictionary, it's a studious inquiry or examination, especially investigation or experimentation aimed at discovery of an interpretation of facts. Now, this is quite a crisp and meticulous way of defining research, as it clearly states that we are trying to examine a specific aspect and we are investigating or experimenting this particular aspect very clearly. The, as per John W. Trussell, it is a stepwise process. Research is nothing but a stepwise process, which is you. It's a stepwise process which is used to collect and analyze the information, and there are multiple steps involved when you are collecting and analyzing the interpretation. It consists of three steps. One is you pose the question. Two, you collect the data pertaining to the question. And three, you answer the question very clearly. So these three steps, posing, collecting, and answering the question, are critical in research. Now,
Another significant aspect which is involved with research is that research cannot be done randomly per se. It has to be done very systematically and it has to be very organized because, as mentioned, as highlighted here, it involves collection, organization, and analysis of data on a specific topic. Now, this is very essential that it has to go in a methodical manner like this, that you collect, you organize, and you analyze the data on this specific topic. Now, and a research in any particular field is done in order to improve and discover our existing knowledge. Now, we already have certain idea of the topic and the knowledge of it, but we try to improvise it so that it can help us further. So it is done to expand the scope of the knowledge in a given particular area. Now, first you have the existing data which is available. Based on the existing data that is available, we create experiments. We first frame the hypothesis, then we create experiments, we formulate procedures that need to be done, and collect the complete available data based on the experiments and procedures, and we reach logical conclusions. So basic research is aimed at improving the existing knowledge which could help in betterment of human race. And ultimately, at any given time, it is important that we are doing research for this main purpose, that we can progress further and help everyone out by getting out better methods of doing the whole process. Now, having said, giving you a brief introduction about research and what it is and why it needs to be done, let us come to the aspect of different types of research. There are three types of research which are essentially followed. One is qualitative research, second is quantitative research, and third is mixed research. Now, when we say qualitative research, it's all qualitative data. We try to collect the data, analyze it, and just present it out. Quantitative research, on other hand, is extensive. This is a kind of research which most of the people do in their thesis and they work out in their thesis. Quantitative research extensively involves the experimentation and the various data collection points, and based on which you draw the conclusions. Then we have mixed research, which is a combination of both qualitative and quantitative research. In some of the cases, mixed research is essential in order to work out both of them. We will be discussing each of this in detail and the methodologies which are required pertaining to this in detail so that we understand and appreciate what are the various aspects associated with each of them. Now, what exactly is qualitative research? Now, qualitative research, as said, it is an approach which involves qualitative analysis of the data. Now, when I say qualitative analysis of data, what I mean to say is we are not doing any experimentation here. We collect the data in the form of videos, images, survey forms, and we prepare questionnaires. So we give these questionnaires to volunteers so that they can answer them based on the questionnaires, which are once the answers are obtained for the questionnaire, then we evaluate the questionnaires and we go ahead based on the evaluation of the questionnaires and conclude based on whatever patterns we observe. So as highlighted here, it is the preparation of questionnaires, collecting the data, and then we go to the pertaining topic, whatever we are talking of by doing all this. Now, this approach is widely utilized in epidemiological studies and studies pertaining to effect of variants in a large group of population. Now, this aspect can be beautifully explained with the help of example which is cited here. We are working out on drug treatment for patients. We are giving a specific drug or combination of the drugs. And when we are giving these drugs or combination of drugs, how does it work in a given population? So here we don't look at a large number of variables we are working on, but we are look at what do you call very limited variables, and that effect of variables is not the one which we look very much. So we frame the hypothesis, and then we use the hypothesis for successfully, explaining the whole aspect as how it is to be done. So if we are using a combination of drugs for the study, then we utilize that combination of the drugs, and we frame questions as how the combination works and how it is going and how we are doing it. And we first frame the hypothesis from the literature. Then based on the hypothesis that is framed, then we do the collection of the data group, and then we go ahead working out on it. So hypothesis is framed and tested in a large number of individuals based on the data and summarized without any specific experimentation. Okay, the qualitative research involves as the voice audibility is a problem. I'm just going back to this slide and trying to explain out what is qualitative research and how it is. So in the kind of the qualitative research approach, you prepare questionnaires, you collect the data in the form of videos, images, and survey form. Now, this approach is widely utilized in doing epidemiological studies and studies pertaining to the effect of variables in epidemiological studies and studies pertaining to effect of variables in large group of population. Yeah, there is a limited quantitation of variables and their interdependence, which is seen here. Now, as you can see in qualitative research, we do not focus on a large number of variables. So it is essential that we look at the overall perspective and we don't enter into too many of the variables and their work. So first we frame the hypothesis, and once the hypothesis is framed, then we go ahead in order to work out based on this. So essentially, you frame the hypothesis, you work out with the hypothesis, and then you try to summarize based on the entire thing as how you work it out and how you do it out. So qualitative research in general does is very essentially it is summarizing in a large group of population as how the things are done and how we work it out. We don't go on into detailed experimentation here, and we look for very limited amount of variables that can be affecting the overall thing. Now, quantitative research is the one which deals with the variable aspects and in details as such. Now, coming to the next aspect, which is quantitative research. Unlike qualitative research, quantitative research is very distinct. We first frame the hypothesis. Once we frame the hypothesis, we have to frame for the experimentation and the methods for doing the experimentation. So we start collecting the data by experimentation. We collect the whole data. We draw conclusions based on the obtained data and summarize the novel observations from the study. Now, that is what is highlighted here, the data by experimentation, drawing conclusions based on the obtained data, and summarizing the novel observations from the study. Now, quantitative research is often very specific. We go ahead and we take a specific topic, and from the specific topic, we work out very clearly the questions and how we have to address them and collect the data. Now, data collection and analysis are from, they can be from a very specific particular set or group of people, or it could be from a large population of people. So it exactly does not matter. But here we analyze each of them in detail, and then we try to study as how exactly we work out each of the experimentational setups. Then you have the mixed research. Now, in case of mixed research, the mixed research is a combination of both qualitative and quantitative methods for analysis. Now, usually the qualitative data, whatever you have, it is a collection from the literature. Once you collect the whole data, that particular data is utilized for identification of the desired topic of the research. So you first collect the data, you frame your hypothesis. Once you have the hypothesis, then we proceed ahead to do quantitative research. Now, one example which can be used here for working out on mixed research is the large-scale genomic studies in population. People usually publish a large number of data on observations on a large scale for alteration of gene expression patterns in a particular or in a group or a cohort of population. So once you do that, you get the so-called metagenomic analysis data. Once you have the metagenomic analysis data, this data is completely summarized in order to raise the significant points. Once we raise the significant points here, then we frame a hypothesis based on the whole thing. And once you frame a hypothesis based on the whole thing, we further go ahead to ask the next steps of the question and how to do the experiments and address them from the available data. So usually metagenomic study analysis, whatever we have in populations, those are the particular ones which help us to give the initiating step, followed by which we do the quantitation, and we finally get out with the whole complete idea of what's going on with a specific trend or a pattern of a gene. Now, having said and introduced to the various aspects associated with the research, how do we go about with the so-called various, how do we go systematically about doing the research? Now, when we are doing various aspects associated with the research, the ones which are outlined here are the most important ones. The first one, which is important, is to identify the research problem. Now, it is essential because we need to know what exactly we want to do. We need to know what exactly is the question that we are trying to ask. So, identifying the research problem is the most important thing here. Now, first we identify the research problem, then we go to do the literature review. Now, the research problem and literature review are interconnected. Now, we do a complete review for the particular topic we want to do. For example, when we have a specific question like a gene, a particular gene or a protein has a role in the overall cellular function. Now, that is the topic which we want to work on. That is what I have in my mind that I need to work. In which case, we know what the protein we want to work on. So we collect the complete literature on that protein. So what exactly does that protein do? How exactly does that protein function? What, what are the various aspects associated with the protein? All these have to be done very clear. So that is encompassed in literature review. Now, once you have literature review, we come to the third step, which is purpose of research. In case of purpose of research, from the literature review, we get indirect evidences regarding what we want to do. Now, once we get the indirect evidence, we propose our purpose. Say, as how I have mentioned, a specific protein has function in cellular maintenance. Then, at that case, we collect all the evidences which are implicative or which are trying to explain that yes, this is the case. And so from there, from based on all that literature, now we try to give the main question that we are trying to work out. What is our purpose? So my particular purpose is to understand what is the functioning of this protein in cellular maintenance or in cell metabolism per se. This is just an example to mention. So identification, once we have the purpose of the research, then we go for making a conceptual framework for research. Now, in this case, when we are trying to make a conceptual framework for research, we have the hypothesis, we have the literature review, and we know what is the research statement that we are talking about. So we go ahead and we frame a model for hypothesis, like how we want to do, how we go about with it, and how we further proceed ahead. So that is where comes the research methodology. Now, the research methodology will be very systematic, in which we try to put forth all the aspects we need. Then, once we explain the methods that we are doing, once we explain the various aspects that we are doing, then we give the experimentation part as how we will do and what we will do. Then we go ahead with it. Now we come to the results. So we do the experimentation and we get out with the results. Now, once we do the experimentation and once we come out with the results, then we go ahead and focus on discussing as how it could be discussing on as how the results could be presented and how we could manage to do it out. Once we have the results, then we discuss it out. From the discussion, we make a clear-cut conclusion as how and what is the interpretation we have and how we have done it. So overall, these are the various systematic steps which have to be followed, which are associated with research. And once we have these systematic steps which are followed, then we go ahead and we work out on the various aspects which are there as such. So this is the organized way by which we can go about with the whole methodology, and we will work out to see as how it is functioning and as how it is working. So I will be presenting each and every of the aspect in detail and try to explain as what and how they are and how we go about with it. Now, coming to so this is a flow chart which has been put here and which where we are trying to explain the aspects associated. So we first define the research problem. Once we define the research problem and we identify the topic of the research, we go ahead and we do the literature review. Now, once we have the literature review, then the whole literature review is summarized, and based on that summary, we frame a hypothesis. Once we frame the hypothesis and we work out on the hypothesis, when we propose the whole methodology with the logic which explains the clarity of the research as how it is being done and the way it is being done. Then we have the data collection part where we will do the whole of the data collection and we work it out. Once we do the data collection, then we do the data analysis, then we do the interpretation and reporting of the data. Now, once we have the interpretation and reporting, Then we come to the conclusions and then we match with whether it is meeting our expected outcome or not. So let me explain to you systematically every one of them now as what it is and how it is. Uh, the research methodology, as how I have said it here, is the most important piece of the whole study. It is, as mentioned, a blueprint of research or study. Now, as it is very explicit here when I say it's a blueprint of research or study, that this explains completely as what has to be done, how it has to be done, and it also explains very clearly as what exactly has the researcher thought about when he is doing the complete thesis work. And RM forms the core of any thesis or any paper because it gives complete idea of the research and also it tells about the appropriate conclusions that one has reached. So it is a very systematic investigation of how the research has to be performed. You have to give all the details regarding how you have framed the project, what is the logic behind framing it, what are the various techniques that you are following, and what are the details that you are using for data interpretation. Appropriate research work which is already available has to be put forth very clearly and concisely. And see, it is in essence the soul of the entire work. So you have to present it very clearly so that the logic by which you have done comes out very clearly. And only then, when you can explain it out in such a way, anyone would accept your research. Now, the best way I would say is you are putting your entire dedication and effort in doing the research for the thesis. Then why not put forth our entire dedication and effort in writing the whole process in a very clear and concise way so that anyone understands what exactly is going on and what we are trying to do. And secondly, it should also give a complete idea of what exactly was going on in the researcher's mind. So it is thus a very, very important aspect of the whole thesis and so should be written very properly. Now, research methodology. In many cases, it has been observed that when we say that it is research methodology, people just confuse it with methods. They just say that, okay, research methodology is just putting in methods and we work out methods, so we are done with it. But it is not exactly the methods. It should not be confused with methods. So research methodology is not just methods. Research methods is a subsection of methodology in which you use all the techniques and the associated instruments that you are using for the experiment. We explain them very clearly, and that is what we do. However, in case of research methodology, it is a complete understanding of the thesis as how you need to do it, and methods form only a part of it, but it is not the complete part of research methodology. Then, research methodology similarly is not literature review. As highlighted here, literature review is complete justification. It's a complete collection of the available literature. We collect the whole literature and we try to justify how we are developing the research topic. Whereas in case of research methodology, we just take a small part of the literature and we utilize for justifying as why we are doing it. So RM thus is distinctive from literature and not literature. So many of them, when we say to have or write the research methodology, they just write the literature review and say it is the same. RM thus includes both research methodology, it includes both the methods and literature review, but both of them in a precise and concise manner. And so it is essential to understand that it is mixing of both of them which will be helpful for us in such a way. Then this what has been put forth here is a flow chart for research methodology with the various aspects which are covered here. First, we review the literature. Once we review the literature and we work out the review of literature, then we come down with analyzing and trying to frame the hypothesis or question. So once we analyze and frame the question that is required, we go about in trying to carry out the experiments that we do and how we work out with it. Then we do both quantitative data analysis as well as qualitative data analysis. So when we do both the kinds of analysis, we try to even defend as what exactly it is going on and how we do it out. Then we come down to what do you call justifying the whole thing and getting out data from the whole process. Once we get out data from the whole process, we interpret and conclude it. And once we have the interpretations and conclusions, we will work out appropriately and see how it needs to be, overall justified in a manner. Now, having said that and introduced you to brief introduction on the research methodology, now we go about with the various aspects which are done in research methodology. It includes the following aspects which have been put out here. So we do in a clear-cut way, in a systematic and in an organized manner. First, we have introduction in research methodology. Now, when we talk of introduction, the introduction covers the justification for research work being undertaken. Now, why we are doing it, what is the necessity to do it, and how do we justify that we only have picked up the specific one. Then we come to the methods that are being followed. What are the various methods that we follow when we are doing the methodology? Like what are the various techniques that we use? What are the various experiments that we perform and how we do it out? Based on this, we go ahead and we proceed with the interpreting the data. So when we are talking of interpreting the data, we have various methods for interpreting. The various methods that are involved in interpreting also have to be given clearly and explained clearly. Now, then we finally come to justification. So we have framed a particular hypothesis, and once we have framed a particular hypothesis, then we come and justify whether the hypothesis is being met or not, and if the hypothesis is being met, how it is being met, or if it is not being met. So we clearly mention these aspects and work out on these issues, and so we have the various steps systematically here in research methodology. So we go then explaining each of this and see how to work it out. Now, these are the various aspects that we have to work out when we are talking of research methodology. So here are the various aspects. First, we have the theory part, which you have to work out with and explain it clearly. And based on the theory, we come to the hypothesis. And when we start working out on the hypothesis, Okay, so these are this flow chart, whatever has been put here, represents the basic aspects that is associated with the research methodology. Now, first you frame the theory. Once you frame the theory, then you go ahead and based on the literature review and theory, so we can do the hypothesis. Now, once you have the hypothesis that is developed here, we go ahead further for the observation and testing. So we have the observation and testing, which is done. Then we come for the confirmation based on the interpretation that is done. We confirm or reject the particular hypothesis. So this is the overall flow chart, which explains as how it is to be done and as how it goes on. Now, now I would be explaining each of them in a clear manner, and then we work it out as how the RM gets done with. Now, here are the, yeah, here, for example, we will start now based on the just research method and the various steps that are involved in research method. We now proceed ahead trying to explain methodology for each of the methods that is there. So first we will present the details of the qualitative research and how the research needs to be done and how we have to proceed ahead. So for qualitative research or for any of the research, qualitative, quantitative, or for mixed research, the major aspects associated with these are the three of them which are mentioned here. So first we need to justify for the research approach. Once we justify for the research approach, then we go ahead with trying to talk of from where we got the source of information and how we have obtained the research approach. Based on that, we talk of development of an appropriate model that has been there. So all the three aspects we work out one after other, and I'll explain to you clearly as how they are and what they are. Now, so for qualitative research, as said here, here you have the qualitative research. So you have to first have the justification for the particular research approach. Then we have the source of information, and then we have the final model that has been developed. So in a nutshell, these three have to be done first. Then we develop the hypothesis, and then we go ahead in explaining the entire thing very clearly. So these are the major aspects. Once we have till the hypothesis, these are the major aspects which have to be considered in the part 2, which includes the what are the various methods that we are going to follow. We have to mention it out. Then we have the study design as how we do it and how we work it out. Then we have the crisp and clear hypothesis aspects. We talk about the sample area, study participants, data collection, data analysis, interpretation, statistical analysis, and for any of them, we have to talk about ethical considerations. For any study, irrespective of qualitative research, quantitative research, or any of them, it is essential that we have to talk about the particular ethical considerations, because if we don't have ethical considerations, then in that case, we cannot proceed ahead working out with it, because your thesis ultimately will be landing in trouble and will not be considered. Now, first coming to the research approach. The research approach should be brief and it should be concise summary that needs to be mentioned with appropriate references. For example, we are talking of utilization of combination drugs to cure an infection. When we are talking of utilization of combination drugs to cure an infection, first we collect the data about this from appropriate literature. When we have the entire thing from the literature that has been developed out, then we develop the proper approach. Usually in the qualitative research, you don't worry way too much about the variables, because variables here are considered as a minor aspect, and we do not consider too much influence of the variables, and we need to give an appropriate justification about the variables and how they affect when they are very critical in some of the qualitative research data. Now, various examples corresponding to these I would be coming out at the specific stages when I'm talking of each of them and I will be explaining it to you clearly. Now, here, when we have the variables and we are talking of the extent of influence of the variables, now you have to have what kind of variables are they and how they will influence and how they will affect it. Now, for this, I will give you one example in order to understand. Now, as I have said in my previous work also, if we are working out, like for example, we are doing a combination drug therapy for treatment of HIV/AIDS in a group of population, then in that case, we need to consider whether people are taking the drugs. Are they interested in taking drugs or are they not interested? Now, if they are not interested, what are the factors which are responsible for this? So the treatment is dependent on several variables like the drug usage, how you sell, and then the financial burden, which is how much is the cost of the drugs. What is the stigma associated with usage of the drugs? Are people really happy to use it? Because when they are trying to use it, at that time there is a problem for people, that there might be stigma, that people will come to know that there is a particular, what do you call, infection in people, that's why they are using it. So is it the stigma factor, is it the financial burden, or is it the availability of the drugs? These are the ones which are, what do you call, to name a few, but you can have a large number of such which could be, considered and taken care of in while doing the qualitative research approach. The source of information, as mentioned here, is the research approach, and you has to give rise to a clear hypothesis, and then you should collect the complete literature review. Based on the literature review, you have to work out clearly as what are the various methods that we need to work on to get out of this. Then what are the various what do you call, appropriate, what are the various hypotheses which are already existing, and what is that which we need as a hypothesis. So collection of the material is very important. So already in the thesis, you have literature review. So source of information in research methodology, when you are putting forth, is easy because it is a subset of literature review and not the complete one. And so you don't need a lot of it, to be written, but just concisely summarize it and make it as a subset of literature review, so that it will be helpful for you in order to understand and justify as how you came to the approach and how you are framing the hypothesis. Now, this particular flow or this particular diagram explains the various kinds of qualitative research methods that are available. Now, it shows that it is exploratory research. And when we are saying that it's an exploratory research, then it's nothing but it's exploration, or basically the same aspect dealt in a different way. So you have surveys, you have polls by which you can collect it, you have interviews which you can use, then you have focus groups, and you have observations based on all of them. Otherwise, you can do case study research from already existing literature review. So, and then you can come to the conclusions. So these have been classified here as primary research as well as secondary research. Secondary research is something which comes from a combination of literature. Primary research is something which comes here from all the work that you do. So I would be explaining some of the aspects associated with primary and secondary research in each of the slides, which will help us to understand how it would be done and how we would go about with it. Now, first, the one-to-one interview. When we are saying that it's a one-to-one interview, it's a face-to-face interview. This every one of us have an idea of. Like the person, a volunteer sits face-to-face with the person who is interviewing, the particular person, or like whatever data we want, we collect it from them. We do an audio recording of the whole interview, and then we grade it when we are doing the recording itself. And based on the grading, whatever we do, we further proceed ahead. We collect the audio recordings, and then again we grade it and we do the analysis. Now, in the next case, this is one kind of qualitative analysis. The second one is focus groups. Now, when you have focus groups, it involves a discussion with group of people and not just one person. So we have a group of people sitting here and we do a focused group discussion. When we do the focused group discussion, they are direct discussions and interviews. They are similar to one-to-one interview, but the only difference is here you have a large number of people. Now, focus group discussions, whenever you have, for example, are interesting because they can be utilized on a large scale. Like when you are working out, for example, what is the fear or the stigma associated with a particular infection that people do not want to tell or people do not want to come out with. So you come out with a focused group discussion where, because it is in-house, people come out and express their fears very clearly, and you can collect the content that you need. Then you have the case study research. In case of case study research, it involves in-depth study of a single case or a group, as has been outlined here. Now this case study research are very interesting in case of medical research. Now you are looking at like proportion of people who are suffering with Parkinson's or Alzheimer's. In which case you don't want to go around with large set of people. You just go with a narrow population or with few people who are like infected with Parkinson's or Alzheimer's. So that's the case studies become interesting. And in some cases, if you can't get out with case study research, you can do case study research from literature as well, which forms a secondary research. Case study it could be a single individual, multiple individuals, or it could be a complete organization as well. Then you have the survey-based methods. This is another kind of methods which are followed in detail in order to grade the responses. So here we make questionnaire forms which are designed and utilized for evaluation of a response. Now we have a series of questions which are framed and utilized for grading the response from volunteers. Now, so we give the questionnaires, we make them fill it up. So the questionnaire forms or survey forms could be in the local language, could be in the national language, or it could be in English, depending on how it is. So the volunteer evaluation forms are collected. Then these forms are graded. Then we do analysis and get out the whole data based on how it needs to be, concluded appropriately. So based on all these various kinds of methods that could be used for qualitative analysis, we work out on all these and then we develop an appropriate model or framework for research. So when we are developing a framework or an appropriate framework for research, first we already have the hypothesis. The hypothesis is based on the information collected. So then we now go to the next step. How do we execute this particular hypothesis to see even if it works and whether it is really making sense or not? So we go ahead to try to develop a model. I have put in one such example in the next slide for public-private partnership. So when I say that it's a public-private partnership based model which we are talking of, so here what exactly happens out is we have the collaboration between government and private sector, and that could be very useful in cases where you want to give it to patients. Drugs have to be delivered to patients at a cheaper cost, and it could be helpful. Now this is the particular model that is being spoken of. So this is a model and there's various stages involved in developing the public-private partnership process. So first we have to plan how to do it out. That's the first step. So irrespective of how you collect the data, how you procure it, you have to plan and frame for the candidates. Once you plan for it, then you prepare. You prepare the questionnaires, you prepare the seminars, and the focus group discussions. What are the various aspects that are required? Then once you have it, we implement it. Implementation in terms of we then make the participants, we make an active list of the participants, and then we work out based on it, and then we collect the whole data. Then once we collect the data, we make logical conclusions based on it, and then we come up if the hypothesis has worked or if the hypothesis has not worked. And so that is how is the theoretical framework made for it. And then we proceed ahead further, ensuring it as how you can go about with it. Now, So having said that, now I would be talking about the various research methods that are followed in qualitative research and how they will be helpful and useful in the whole process. Now, so this is basically are based on the already mentioned contents, which I'm going in a systematic manner in order to explain how it is going on. Now, what are the various methods that have to be followed when you are doing qualitative research? So in this case, it is essential that we first go for you, as mentioned here, a clear and concise summary of the methods has to be provided. So what methods you are doing and how you are doing has to be explained based on the available literature review, because if you are going to follow the same methods, if not, if you are going to follow any new methods, then they'll be based on an already existing template which you are going to have it out. So we have to explain both of them clearly and systematically. That could be obtained from the source of information and from the model that you have as what method you will follow. Now, the method that needs to be followed, it needs to be crisp and it has to be clear. So it has to be given in such a way that, it gives the entire idea of the whole thing in a similar manner. Then comes the next important aspect, which is the study design. Now, when we come to study design, this is a very critical part that has to be understood and explained clearly, because you have to give how exactly you are going to do the study. It should explain the research design that has been developed clearly. Now, what exactly are you going to do in the study? Are you going to make questionnaires? Are you going to make any focused group discussions? In which case, what exactly is the plan? So if, for example, if you are going to work out for either analyzing the patients who are suffering with HIV/AIDS and what is the extent of stigma in them, then you have to make a questionnaire. The questionnaire should have various questions, which includes, do you face the stigma? If you are facing the stigma, what kind of stigma are you facing? Is it that it's a social stigma, it's personal stigma, or it's interrelated, or it is at the medical centers you are facing the stigma? Then all such questions have to be put together in the questionnaire. You have to supply the questionnaires to the participants. And then you have to evaluate the answers. As how we grade and evaluate the answers, I'll explain back as when I'm going to the next slides. Then the hypothesis. So how exactly do we develop the hypothesis? Yes, hypothesis could be developed in different ways. One method, hypothesis is developed based on literature analysis. So we do the whole literature analysis, then we develop our idea based on the various evidences that are available. Then otherwise, we have personal observations. Like I am a researcher myself, for example, and I'm working on the effect of a protein on cell polarity. Then I know what it does and how it does. And I have evidences which point out to this fact that it is how it is. So we go for based on the personal observations, or we go for logical deductions from existing research, which is very close to literature analysis. In some cases, logical deductions from existing research also would mean that we have a group of people who are like scientists who are all like close friends and they are doing similar projects. Then we get all their research together and then we draw a logical conclusion. Or I'm continuing the particular research which I'm already doing, so I frame hypothesis based on. In some of the cases, it's highly based on personal intuition that you have. So all these could be utilized for making the hypothesis, and any of them could be successful. Then there are different types of hypothesis that you have. Now, of the types, first one is descriptive hypothesis. In this case of descriptive hypothesis, here we do not associate the various variables with the hypothesis. We just have the characteristics of the variables and associates with the hypothesis. So, which means that what is that particular variable? What are its characteristics? And does it have any effect on the hypothesis? Then the variables here could be person, organization, or any of them. The next kind that you have from descriptive hypothesis is a relational hypothesis. where we look at the interrelation between the variables and how it affects the hypothesis. For example, if you say families with higher income have higher expenditure, we are talking of many things here. We are saying that they earn high, so they have higher expenditure. So if they are earning high, if and they have higher expenditure, there are multiple variables which will be coming in this. And so we all look at like what are the various expenditures we do, and that is dependent upon what are the how much income do they have. And so it splits out to income versus expenditure ratios, which gets interrelated. So when this happens out, it will become a relational hypothesis. However, in biology and in most of the cases, you have two main important hypotheses called the null hypothesis and alternative hypothesis. Now, the null hypothesis and alternate are the most important ones. So, when we talk of null hypothesis, so you have a specific hypothesis which is developed. Does the expected outcome meet the hypothesis? If it does not meet the hypothesis, it's a null one. So that's represented as HO or H0. That's what has been highlighted here. For example, if I say people living with HIV felt a stigma in form of unsupportive social interactions. So am I meeting the criteria there or not? I'm not meeting the criteria for this, then it is a null hypothesis. Now, in case of alternative hypothesis, it's completely opposite. Now, in case of it, it is opposite to null hypothesis. So we are meeting the criteria that we need. So we have a specific hypothesis and the outcome is expected to the hypothesis. So it's alternate. For example, people living with HIV do not face stigma in form of social interactions. So if that is correct, then it's alternate hypothesis. Otherwise, you will be having it as a null hypothesis. So the question of yes or no is the one which decides whether the hypothesis goes towards null or towards the alternate one. Then sample area will be exclusively depending upon the various details related to the sample and description of the sample. So you have to give all the details, like if it's a particular demographic pattern and a particular demographic part wherein which you are trying to build it out, you have to give all the details which are associated with it, and you have to justify why that specific area has been picked up and why we are only working with that specific area and why not with any other area. Then we have steady participants. So steady participants, we have to pick very clearly and in a very proper manner the nature of participants, why a particular set of participants have been picked up. Is it based on the nature of element or is it based on specific demographic region where that element has been found has to be explained clearly. Now the various the participants or the volunteers along who are infected along with the people whom they meet with, this medical professionals and also the stakeholders who are involved in this project who come into this whole thing have to be also listed very clearly and very properly. Then the data analysis is one of the important parts here. Now it has to be first of all the data has to be collected properly and organized properly. Now the data obtained from group discussions and surveys, you have to put it in Excel sheets. Once organized, you do the thematic analysis so that you can obtain logical information. So the key patterns from the interviews are obtained based on the analysis, which will be useful for logical conclusions. Now, for most of the qualitative analysis, it is the statistical analysis which is performed and forms the core of it. There are a large number of statistical softwares which are available that could be utilized. Examples of such softwares include EpiInfo, SPSS, and Stata Interpool version, which are highlighted in bold in the slide already. So we use this. EpiInfo is routinely used. Now, EpiInfo is routinely used software in epidemiological studies, wherein it's used to organize the volunteer data in the form of Excel sheets. The whole thing has to be put out in the form of Excel sheets and we work it out. Then SPSS is used for complete analysis of data and in drawing the charts and to get the appropriate conclusions. Another one that you have is Stata 7, which will be helpful in complete analysis and in writing of conclusions appropriately. So we use any of these existing softwares and then we work out based on this and we ensure that we get out the proper analysis done. Okay, However, for the qualitative analysis, whatever we are doing, it is essential that we have ethical considerations. Now, this ethical consideration goes through for all of the presentations and not just for qualitative. So, informed consent from all participants prior to interviews is very essential. Confidentiality of information, the whole study is essential. There should be no breach of information. This written consent has to be taken from staff as well as from all the people who are handling the data. The various observations, data correlations, and overall conclusions of study have to be kept confidential. So it's like these ethical consideration forms have to be filled up and signed by any and everyone who are involved in the whole process. So having said that, once you have the hypothesis developed, you collect the data and you analyze, you have to conclude. The conclusions have to be like proper interpretations. So first we conclude and we get out with the final statement. For example, a particular drug that is being used for the treatment has effect on the condition. Like I am trying to cure cancer with trastuzumab, then trastuzumab has effect on cancer. So if the final outcome says it has the effect, then the alternative hypothesis has worked. If it says it has no effect, then the null hypothesis has worked. If alternative hypothesis works, it's fantastic because it helps us to publish the results and get out with novel observation. If it is null, then you have to justify as why it is null and see if you can then tell people how as how exactly can it be published further. Now, having said that, that is a nutshell summary of qualitative research. I'm going to proceed ahead and now start presenting about quantitative research, the major aspects associated with quantitative research. Here again, as like the introduction, the research approach, the source of information, and development of appropriate model play a key role. Then, of course, when we come to the major aspects for consideration, it's the study design, which is important. But however, here it will be much less when compared to qualitative research. We have the hypothesis. It differs from qualitative research in the sense we have to explain in detail what are the instruments and methods that we'll be using for measurement, what are the various experimental controls, experiments that you'll be doing, the controls that you'll be having, and how you'll be manipulating the variables to get the optimized data. And then the collection of empirical data and analysis for the same has to be explained very clearly. Now, this is a flow chart which explains about how we go doing it. We first have the theory, which we work out for it. Then once we have the theory, then we go for the hypothesis. We frame the hypothesis, then we do the research design. Once we have the research design, we go about with how to work out on the operational design of the research design for the proposed hypothesis and how to collect the data. So the various steps pertaining to this are presented here. Then we do the data collection, then we do the data processing, analysis, then the findings, conclusions, and the results. And this is like a loop because already based on the existing results, we propose the theory. So it continues in this way. Even for further research to be done, based on one we have already done, we could follow this loop. Now, the research approach is essentially the same as like the qualitative. So I wouldn't be spending too much time on these slides. It's a brief and concise summary of the entire thing. You have to give the appropriate references as well. As for the example here, I say like the Parkinson's protein has effect on viability of cells, just for example. Then what is Parkinson's protein? What are the various functions that Parkinson's protein has? What are its role? Are there any evidences that it has effect on viability of cell? We have to collect all this. Then once you have the entire literature which you have, then you come down based on the implicative evidence that probably it is involved that Parkinson's protein has effect on viability of the cells. Then Yeah, so once you have the summary of the observations, you proceed ahead and work it out. Another way, similarly, an example here is like a particular biomolecule, like a natural flavonoid that is found in the cells, has effect on neuroregeneration. So we search the literature, we compile the entire data, and then we predict whether it's a neuroregenerative agent or not. And if it is, then we proceed ahead to work out with it. The source of information, again, it is not literature review, but unlike that, it is a clear-cut subset of literature review which you have. So it needs to be clear and it needs to be, succinct, in which case it has to be worked out with. So we have to give the whole details in the clear manner, and then we work out based on it. And so it is an approach which will be a subset of the literature. Here is the research methodology with the design and development that is shown here. So first, you identify the problem, you define the objectives of objectives, here in which you start testing out your for what hypothesis you could be making. Then you design and development you do for it. Again, you test and see what experiments can be made out with. Then you exhibit it in the form of experiments, you evaluate, and then you communicate. Everywhere you have to first propose and you have to test the hypothesis, collect the reviews, collect the data, and then you do the interpretation part. So then we have the appropriate model development that needs to be done. So the framework for research is what it is. There's a clear part, as always has been mentioned. It involves that you have to develop a proper hypothesis based on research approach and information collected. One such example, like I will elaborate here, is the effect of drug metabolism on various parameters, which influences the outcome. Like we are trying to develop a drug which can be used in humans, so we have to do human trials. But for that, we have to start from the beginning as how to synthesize the drug and how we proceed with it. So I have a outline in the next slide which shows and tries to tell you the extent of analysis that needs to be done here. Now, for example, as you can see here, first we have to see the solubility of the drug. Then we have to see whether it binds to the appropriate receptors and the potency of the drug. Then we have to do pharmacokinetic studies regarding its availability, its distribution, its excretion, and various other parameters, including its safety. Then we do the PK studies, the dose exposure studies, and then we come to the safety profile. So if you want to use the drug, then whether it is safe to be used or it is not safe to be used. Then we go for the safety efficacy models, the trial simulations, and the phase 2 trials. After all these, we finally come and put it for approval with the drug agency. Now, once we have the approval from drug agency, we have a marketing strategy. This is a complete flowchart as how you can develop for the drug, and that is how is the in-depth analysis which you need to have if you want to work it out. Then we have, then we have here the study design which needs to be done based on this. So the study design should explain the various methods that you are going to follow and the various instruments that you need. Now the various instruments that would be used for study have to be explained. Various reagents that would be used for analysis have to be outlined. Various experiments that would be done need to be explained clearly. Now, as said earlier, the hypothesis could be from any of the aspects. The source of the hypothesis could be from literature, from personal observation, logical deductions, or continuation of existing research, based on which we have the hypothesis developed. Then you can either have the null hypothesis or you can have the alternate hypothesis. Null would mean that the proposed outcome doesn't match with the hypothesis that is framed. Alternative is opposite to null hypothesis. Then we come to the methods that have to be done in quantitative research, the instruments and methods for measurement. Now, you have to identify the appropriate instruments that are required for analyzing the samples. Now, we need proper measurement methods here. For example, a particular experiment is being performed where one wants to measure glucose estimation from blood, and we have a large number of samples. So we need to identify what are all required for estimation. First, we will be needing what do you call it, glucose assay kit. Two, we will be needing to a spectrophotometer, because the end point will be a colored product which are read on a spectrophotometer, and we need to draw standard glucose curves. All these have to be kept in mind, and appropriate you have to do. Now, in this case, this particular aspect is for a single experiment in which there's only like measurement of glucose. But however, if you are talking of complex scenario in which you have got multiple things have to be measured, then we would be needing it out to be done in a different way. For example, we are talking of genes which are involved in a metabolism of an organism, and if they are like multiple pathways and multiple end products, in which case we have to list out for each of them, so the intermediates and end products, as how we will be using them, as how we will be assaying each of them, and if the assays don't work, what are the alternate methods. One of the examples for alternate method which is proposed here is many of the enzymes involved in the pathways bind metal ions, and hence they have color. So we could do so-called colorimetric protein readings or fluorescent protein readings using probes to see that whether the proteins are functional or not, and hence assay out if that particular pathway is existing or non-existing in the cell. Now, yeah, they are now all the experimental details which are required have to be given very clear. Like the various experiments that need to be done have to be addressed clearly. What exactly are those experiments and how you are planning to give. Then various positive and negative controls needed for experiment need to be framed properly. The samples required need to be collected and stored at proper temperatures. So you have to mention if they are temperature specific experiments, how you are processing the samples. For example, if they are certain samples which have to be subjected to cryo-electron microscopy, the samples have to be stored at minus 80 degrees and they have to be cut ice cold so that they can be provided in that way. So we have to give all the related details, and we have to explain all the related details in a specific manner like that. Then, one example which has been put forth here for the experimental details for identification is this. Now, this particular example deals with identification of differential proteins that are expressed in different cells. So here we have the cells which have been shown here. You collect the various cells, you make the protein from them, you do the peptides by proteolytic degradation and analyze by mass spec. Once you have the mass spec, you have the raw data, you do the database search, then you do the pathway in which they are analyzed. So, and we can draw differential expression. We can draw for differential expression of the various proteins that are present here as shown here. Like some proteins are specific to only one cell and not to the other one. And when we have such a this thing present, we can use that as markers for identifying those cells. Then we develop appropriate assay methods for detecting only those specific proteins. Yes, then another example which would be given here apart from this is we want to do commercial production of surfactants. Surfactants, as you know, are like very useful for removal in day-to-day life of lipids and oily substances. So the commercial production of the surfactants, if you want to do at a large scale, first we need to check out the variable parameters and others. So for that to be done, we first have to identify the starting material from where you will be making the fatty acids. And the fatty acids made have to be analyzed for their various physicochemical properties like acid value, saponification value, total fatty acid content and others. And once you have those being there and those being done, then we proceed ahead to the next step of identifying and further making the surfactant. Then we make the surfactant by appropriate method. Then we need methods like NMR, IR, and GLC, which have to be utilized. and used for characterizing the surfactant. You do the physicochemical properties of the surfactant in terms of its wetting time, foaming properties, cloud point, and crack point. You also look at the stability. And once you have all this done, then we release the surfactant into the market. So this is how are the details which have to be done. So this is just one of the examples given in order to highlight the various experimental details that have to be produced and that need to be done such that it works out in an appropriate manner. Now, when we are talking about quantitative experimentation, we have variables. There are so many variables which are present when we are doing here. The various variables that could be obtained here or done could be processing conditions for a sample, analysis conditions, and conditions specifically utilized in the assay procedure. Hence, all of this have to be manipulated effectively in such a way that ideal experimentation method is established, which could effectively help in optimizing the whole procedure. So we have to do the manipulation of the variables carefully so that we get the optimized method. And once you have the optimized method, then we work out. And get out the whole data. Now, one example here as shown is for this particular one, for the variables is shown here in this particular pictorial representation. When we say that they are variables, they are multiple variables. Some of them are dependent, some are independent, and some are controlled. For example, we are asking a basic question like, does red light affect the plant growth? You want to answer. Then you expose the plants to light. The independent variable here is the color of light. That you cannot, you have to put only for the red light. Whereas when you are talking of dependent variables, one of them is the plant height, which comes because plant height can be controlled. And the ways you control it is how you water the plants, what kind of soil is being used, how you are controlling the temperature, and the day and night control. If you have all this, then in that case we have the various control variables that are done with, and then you have the whole process being worked out. So all these have to be looked up. So this is used as a specific example only, but all of these have to be worked out only when we can look up at the variables and try to have conclusions based on the variables. Now, we go for, once you have the instruments, once you have the experiments ready, we do the experiments along with the instrumentation, and we collect the empirical data. So the data collection is based on the observations obtained by performing the experiment. That is what is the key point there. Now, the various observations obtained, irrespective of whether they are major or minor observations, have to be recorded properly and they have to be documented properly. Now, the data can be collected either by qualitative methods or by quantitative methods. When I say quantitative methods for data collection, here it means accurate numbers pertaining to the experiment. Each and every number that is recorded for the experiment has to be put forth and has to be used. Based on the obtained numbers, you have appropriate interpretations. However, in this kind of quantitative methods, you just go for the data. You don't worry too much about the quality of the material. Now, one simple example if I am giving here is, if you are counting number of apples which are falling from the tree, the quality of apple doesn't matter. We just see how many apples are falling. That's like a basic example which I have given here. However, if we are going for qualitative interpretation of data, then it is a different aspect. Qualitative interpretation depends on the quality of the material and what effect it has. So in this case, what happens out is, when you are talking of the quality of the material, for example, we are shipping any of the consumable products like grapes or apples from one particular place to another place. In that case, whether the complete set of apples, like for example, I have put in a kilo of apples shipped from here to Delhi, then have 1 kilo reached correctly? Are all of them consumable? If all of them are not consumable, then the quality of material has been severely compromised. So then the qualitative interpretation of the data becomes very important at that point. Now, analysis of the data. Now, analysis of data in many of the cases, it's not just collecting the experiments, having the data, and just concluding from it. It has to be analyzed thoroughly and properly. You need what are called as controls when we are doing an experiment. You need both positive as well as negative controls, and we have to analyze carefully. Only upon, yes, well, so we take a particular experiment, we use the positive controls, we use the negative controls, and then we confirm the validity. First of all, if the experiment has worked. So if the negative controls are fine, positive has worked as expected, and then you are getting results. So which means that everything has worked fine, and that particular experimental set can be considered. Otherwise, it cannot be considered, and it has to be discarded. And if we have such discardings, we have to repeat the experiments multiple times to ensure the accuracy of data even before you do it. Then, while analyzing data, you have what are called as false positives and false negatives. False positive is you have a false positive data is a one there in which you are getting the expected outcome, but that is judged wrongly when you're doing it. The test result, it's an error in which the test result incorrectly indicates the proposed outcome. For example, for a simple example, a pregnancy test shows that yes, the girl is pregnant, but however, it is not positive. She is not pregnant. So there's some flaw which is associated with it, and not because of the test. The test is the problem, and not with the what do you call the pregnancy related part. False negative, on other hand, is opposite to false positive, and here the girl is pregnant, but the test shows that it is negative. So we need to use other methods. So this is what is examples for these two, and we have to consider both these aspects when we are talking of it. So it's called the false discovery rate. It tells the extent of false positives or false negatives in a given experiment. Both have to be utilized thoroughly, and then you have to eliminate the experiments which don't meet the requirement. After eliminating the errors, the final data needs to be obtained and utilized for all the work that has been done. Now, how do we do modeling of the data? Now, the modeling of the data, as has been said earlier also, the Excel sheets are one of the most beautiful ones which are helpful here, and that's how we go about with it. Most of the data collected is subjected to analysis using standard analysis available in Excel sheets. Standard curves are drawn and regression analysis done with slopes and intercepts are obtained. So you can also have calculated the students' data standard deviation and averages, which are the normal statistical parameters for which Excel is more than enough. However, if you are doing like a more serious form of modeling, then you would be needing advanced software. So the major ones which are used here, other than Excel, are GraphPad Prism, R, SPSS, SAS, and MATLAB. MATLAB and SPSS are most widely used because they also have chemical simulation software. So you can simulate fermentations and others, and you can use it out in easy manner. So you have the Excel most widely used, followed by the GraphPad Prism. Now, when we are doing the GraphPad Prism, it's a routine way of getting out the ANOVA data and accurate standard deviations which are there. The software is also useful for making graphs for animal experiment studies and is highly user friendly. Excel, though used, all features are not present. So that's why you will be needing the GraphPad Prism for analysis. And as said earlier, here again, you have ethical considerations which need to be considered and which need to be there for the entire part. So informed consent from all participants is required. Confidentiality of information is essential. Then you need the return consent, and the various observations, data correlations, and conclusions are confidential. Because though it's a quantitative study, it doesn't mean that you don't maintain confidentiality. You don't want your thesis data to be out for everyone till it is published as papers and till it is accepted thoroughly, so that there can be no plagiarism of the thesis that is associated. So having said that, uh, once we have the data, once we draw the conclusions, and once we draw the interpretations, we go ahead for the hypothesis, which includes the null and the alternate hypothesis. Now, if the particular our expected outcome and the hypothesis are matching, then its alternative is perfect. We can go ahead and publish the data. However, if it is null, then in that case we have to think of why it is null and what exactly could be done to defend the hypothesis, and can we go ahead and publish it out, and how it will be done. Now, that with that, we cover the various methodological aspects that are associated with quantitative research. We come to the last and the final section, which I will be covering for some more time, is on the mixed research. So when I say of mixed research, it's a mixture of quantitative and qualitative data. As can be seen here, you have the quantitative data obtained with some experimentations. So you look up at the literature and you get the qualitative data. So you get the data collection, data analysis from this set, you get the data collection, data analysis from this set. And then we put in together, then we compare the results, integrate and interpret the whole thing. Now, when we are saying of mixed research, we are mixing the data. So when we are mixing the data, how is that we mix the data? So when we are mixing the data, it could be done in any ways. You merge the data as shown here. One, we have the qualitative data. So you pick up the qualitative data. Then you have the quantitative data. You pick it up. You mix both of them and make the final results. Or two, you have the qualitative data. From there you frame the hypothesis and connect quantitative data. And once you have the quantitative data, you come to the final results. Or it could be merged together, that the qualitative and the quantitative can be together as such and could be clubbed to form the results. So it goes, it is up to the researcher who is doing it as how you can club them and how you can do it. You embed it or you do it systematically. That is up to you. Now, when you already have quantitative and qualitative research which are available, what is the exact necessity or purpose of mixed research? Now, so there are certain places or areas in which mixed research needs to be done. Now, if mixed research combines the strength of both, combines the what you call the strength of both qualitative and quantitative methods and overcomes their weakness. For example, when we are doing qualitative data, we don't do much of experimentation and we just propose. In case of quantitative data, we don't look at a large scale. We just look at only the defined one and we go ahead with it. But so that weakness of not looking at the large scale and this weakness of not experimenting with a large scale, when mixed together, will be very helpful to give you the best possible question that you could use and answer it successfully. Now, the rationale for using the mixed method when you are writing the methodology has to be provided clearly. Why you can't use a single method, why you have to go ahead and use two different methods has to be provided, mix them together, has to be given clearly. Only then and when it will be understandable. Now, there are two models when we are doing the mixed methodology. One is the sequential mixed model and other is called the concurrent mixed model. Now, when we have the sequential mixed model and concurrent mixed model, how we proceed ahead with it, I'm explaining it next slides. Now, this is the sequential mixed model. Now, you have qualitative and you have quantitative. So first we do the exploratory phase. We identify the purpose for the research, then we do the data collection, analysis, and inference. This inference is used to frame the hypothesis and you go for the purpose, which forms the purpose here. The inference becomes a purpose for quantitative research. Then we collect the data, analyze, and infer. You can mix both of them to get the final meta interference, or it could come from the other. Now, in case of concurrent mixed method, In case of concurrent mixed method, you do parallel both of them. Like you have a purpose which is framed from either qualitative or quantitative. You individually collect data from qualitative methods and quantitative methods. Then you analyze the data from each of them separately and you mix them together and do the final inference. This is called a concurrent model, which is utilized per se. How do we frame the hypothesis in case of mixed model? This is a very interesting question. The development of hypothesis can be done either by quantitative analysis or by quantitative analysis or vice versa. So we have here any of the ways can be used and it is left up to the end user as how he wants to do it. First you frame the question. Whether you frame it by quantitative or qualitative methods doesn't matter. Then this hypothesis again framing can be based on personal intuition, available data which is pointing towards specific observation, or based on indirect evidence. Irrespective of any of them, you frame the question. What are the methods that you follow in case of mixed? How do you do it out? So it will be a mix of methods that are followed for both qualitative analysis and quantitative analysis. So qualitative analysis in form of group discussions, case studies, or any other relevant methods, you collect the data, you assess it, you interpret it, and logically conclude. Then once you have so all the softwares and the various other statistical analysis done, you finish it off, you get out the interpretation from qualitative. Once the interference is obtained, then you frame the hypothesis and you go to quantitative analysis. Now, based on the hypothesis from the qualitative study, you frame appropriate quantitative analysis. So, what are the experiments to be done? What is the sample size? What are the assay methods? Then you get out with various variables and the various, optimization of the variables to get out with the best data. Once you obtain it, then you come to false positives, false negatives, and non-reproducible data, and resulting in generation of highly validated data. Now, this can be seen whether it's matching with the null hypothesis and whether it is matching with alternate hypothesis. Now, this is the mixed method strategy as it is shown. Do the quantitative, do the qualitative, then do the quantitative. When you come to the data interpretation analysis, then you can bifurcate. You can have data and conclusions from the qualitative part as well as from the quantitative part, and then mix both of them together and give the final integrated interpretation, which will be a combination of both of them together. Now, having said that, I will be providing you an example of how exactly we go about and do this and what is the method of doing it out. So here we have one particular aspect that would be covered here is the quality. Here is the techno-economic considerations for commercial production of organic solvents. So when we want to produce organic solvents at a large scale, we need to have different steps. So, but first, before that, we have to do the pilot scale study and try to understand as how and what it is. So, first step involves the various qualitative methods that are required for estimating the yield for extraction process. So, what is the raw material that you are going to use for extraction process? How you are going to do it in silico by using various softwares. So, softwares such as Aspen Plus and MATLAB can be used for simulation of fermentation. So first you start with the raw material, you simulate the fermentation and see how you are getting it out. Once you get it out, then you look at the various downstream processing methods and see whether you can get a clear solvent or you are not getting a clear solvent. Now, the various methods that have to be used here, you can get from literature. And those methods, in fact, most of these commercial softwares like Aspen and MATLAB have already methods from literature review. So we go ahead with it and we feed those commercially available options in the software, and then we see how you get for the downstream processing. And once you have the downstream processing which is done there, So we have to next consider is the feasibility of producing this, because they are organic solvents, they are sensitive to temperature, and they are losses which are associated with it. So we have to think of the thermodynamic constraints that have to be effectively checked in and based on these conditions, on the optimized conditions. So what are the various thermodynamic constraints that have to be used and that have to be developed? Then we do stimulation studies are done for evaporation and for purification methods. So based on the stimulation studies, we get an idea of the sensitivity of the process, what is the uncertainty associated with the process, and sustainability of the process. So when we have this, we go for the commercial considerations, the economical considerations, or the feasibility for working out this particular part. Now, capital cost is one of them which has to be considered, because this will include so many equipments for fermentation. It will include the raw material for fermentation. So we have to consider as how exactly we go about with the equipment cost, material cost. Is it feasible to buy those and do it? Then what is the cost for operating the whole setup? Then we talk of cash flow analysis. In terms means, if I'm having this setup and I'm able to produce it, what are the monetary benefits that are expected based on this investment? Because the plant over time has depreciation, so we have to consider the depreciation values as well, and we have to explain all these in detail and say what it is and how it is. All these can be done using simulation software for fermentation. Based on this overall analysis by qualitative methods, once we have identified the perfect method for production, then we jump to real experiment and then we do it. So that this method, finalized method, will be useful for production at the industrial level for production of solvent. So that is one of the examples which I have given for the mixed research. So thus I have tried to cover here for the various aspects of research methodology related to qualitative research, related to quantitative research, as well as to mixed research. So thus the research methodology is a significant milestone in thesis studies. If you write it clearly and you write it concisely, it will give you the entire logic behind the research work that is done. A well-written research methodology will give a clarity of thought process by the researcher and will be very helpful for understanding the whole process as how it needs to be done and how exactly can you work out on the whole process. So, having said this, so having said this, I then conclude my presentation. We will be happy to answer your questions in the chat box. Thanks for attending the webinar, and I pray that every one of you stays safe and then you stay healthy. So now the questionnaire session is open. You could go ahead and you can ask me for any of the questions that you want to. Okay, there is a question from Srinivasa Padmakar regarding the data collection process at field level through questionnaire during COVID situation is very difficult. So many organizations are not giving information saying that it's not confidential. How alternative of data collection can be done in Google Sheets? So here, the question that Srinivasa Padmakar asked is that during the COVID situation, it is difficult to do in field. So how is it possible and how we can work out on it? Yes, one idea that you could do is you collaborate with the hospitals and get out the Excel sheets that are available. And once you have the collaboration with them and you can get out the Excel sheets, you can collect it out and you can proceed ahead working out with the entire thing. There is a question from Dr. Naila Yusuf. Any research, how many literatures are reviewed? There is no such cap on how many literatures are reviewed. When you are reviewing the literature, it is very important that you put whatever is appropriate to your topic. Don't try to put in the entire thing, say in terms of secondary research which is presented and all. Try collecting everything that is possible from your side. And once you have everything that is possible from your side, then only we would go and then we would finish it off. So there is no cap, but however, that has to be decided from your end based on how specific are you collecting the complete set of questions, papers related to your question. Now, there's a question from Diksha Singh who says, is there a limit for minimum number of reviews in the literature review chapter? That answer depends upon where the thesis is being submitted. Like overall thesis, the number of references that you have is capped in. So in which case you have to look at the the organization and how they are saying what it is and how it is. Then you can. Usually literature review has maximum number of references anywhere between 70 to 100. But please check with the appropriate organization or the appropriate concerned university regarding the rule if they have on it. Okay, there is a question from Dr. Mahesh which says blood glucose level metabolism example for science. Now, I had already mentioned regarding this that, yeah, you are trying to monitor on a large scale the blood glucose level in a population because you suspect there is a large number of people who are having diabetes. Now, if that is the case, that is a particular aspect you are considering here. So you can't do spectrophotometric assay every time. So you can just use the blood glucose test strips, see the color visually, and you can confirm what it is and how it is. And based on the strips, it will become completely qualitative without involving any experiment then, because it's based on the color and you're not going for spectrophotometry. There could be sort of deviations which you can have, and hence you will stop it out there by the color based one. You take the images, then you grade them, and then you use it. Yeah, there is one aspect from Kabita Sahu. She says that little bit discussion on health research. Health research is a broad term, and so I didn't involve much of it. But yes, if you are talking of epidemiological constraints, for example, like you have a particular one, like I can take an example of Ebola fever, which infects people specifically in the African Congo region. That is a specific area where in which it happens, and that's a very specific way in which it happens. So we can collect data from there by participation, by questionnaire, by survey. Actually, you don't need to do it there because you'll have the complete data in Excel sheets. You can have collaboration with the hospitals. You can collect the Excel sheets and you can analyze the data and you can get out with the key patterns that are there. So once you have the key patterns which are available and which you work out on, and you can proceed ahead and you can publish it in collaboration with the hospital itself. So in that way, it would be useful. There is a question from Mrunalini Rana. She says, how is objective of research and hypothesis correlated? That's a good question. Now, objective of the research tries to tell you justifying your topic. So that is the objective, that's the main objective of the research. Whereas hypothesis correlated in the sense means you have your objective. Based on the objective, you are framing the hypothesis. So they are interrelated. So when you have the hypothesis which is already there and it's framed on the objective, if the hypothesis is not true, you have to go back to your objective and redefine it so that you will have an appropriate answer and an appropriate explanation for the entire thing. In that way, both become highly correlated as such. Well, Dr. Mahesh has a question on social research. That is an interesting one, but however, uh, it's a different field in itself. So it's definitely worth considering it. But then, social research here will be altogether different. But I can give you some sorts of hints which I have presented in the presentation already, like the stigma factors and the associated with a particular infection that comes in this definitive social trend, because it has to be worked out at the society level, at the individual level, and at the interstitial level between society and individual, with the various ones you're talking to and you're working on. And hence, it becomes very essential that you have to dissect each of them out and you have to work it out. In that way, in this presentation, I just put in briefly that part of it, but that you can enumerate it out much more, and you can use it for working it out. Huh. There is a question from Vijay Kumar M, which says, how to compare different qualitative method has to be chosen for a particular research topic. Now that's interesting. You have so many qualitative research methods which are available. So the comparison of different qualitative methods and the right one to be picked up will depend on your research topic. Pick your research topic, you have your question. Once you have your question and you have done the research topic, then we go ahead with it and identify the various methods that are to be done and that have to be followed. And once you have that out, and once that has been worked out, then from the methods you get to know which are all the best ones which you can use and how you can go ahead with it. There is a question from Lalitambika Devi, LS, and she says role of design in research. In fact, that's very important. If you cannot design your research properly, you are not able to show the exact aspect related to it, then it will not be crisp and clear as how you have done it. So the design is very important part, how you are going to do your experiment, how you are going to manage to get it done. What are all the various things that are required for it? You need to explain very clearly. When and only then it will work out. Otherwise, it will not work out in any ways. So you have to appropriately do it out in that way. Yeah, there is a question from Vibhor Avasti, which says how to check reliability of questionnaire. Now that's an interesting one. Now the reliability of the questionnaire will be depending on first how you frame the questionnaire. Once you have framed the questionnaire and the questionnaire has been answered, you will understand whether the particular volunteer or participant is just doing it for namesake or is he seriously answering it. Now, if he is not serious, so you can, if the questionnaire you are not able to trust, call for a one-to-one interview and check out as what it is. Then you will understand what it is and how it is, and you'll get to know it. There is a question from Suvarna which says how to do interpretation. Now, interpretation has to be done based on the results. First, put in the results. If you are not understanding how to interpret, go back to literature review and from your purpose of the research, study what it is, how they have interpreted it, and you can follow a similar model for your interpretation. Now, there's a question from Swarupa Chakravarti which says, what is the basic difference between qualitative and quantitative research? Now, that's what I have highlighted in the presentation as such. You are just looking at the accurate numbers. You are not looking at the overall quality of the experiment, then it's quantitative. But if you are looking at the quality exactly, and if the quantity is not the one important, whether the quality is there or not, then it's a qualitative research method. Now, there's a question from Nidhi which says, how can we learn statistics tests effectively? I'm not able to get proper media. Well, you have online free demo classes which will tell you how to do it. So you can use it. For example, if you want to do GraphPad Prism, there is an online free server which will be available only for 20 days after downloading, which you can use and you can help it for conclusion effectively. So you can learn how to do it out. Once the free one expires, you can purchase it and you can learn it out. Yeah, there is a question from Vijay Kumar VM. How to do comparison of different qualitative methods chosen for a particular research topic? Yes, well, I think I answered this question earlier. It will depend on what kind of the topic you're doing, how you're working, and then we go ahead with it. And based on the methods, we decide how to do it out. Yeah, there's a question from Elena which says, how should literature review be structured? Yes, the structuring of literature review is slightly complicated because everyone feels as though it's like you just keep on writing because it's a literature review. But unfortunately, it is not the way. So in the literature review, you have to frame what all you want, what is that you are trying to look for. First, give idea of what you are looking for, then give idea of what that particular thing does. Then you come to the conclusion and frame the appropriate question. There's a question from Ranjani Rajagopal which says, how many pages of thesis is expected or accepted for PhD? Well, there is no definitive answer for that because it varies anywhere from 100 pages to 300. But bulkier thesis as you start writing becomes more of a problem. So try to keep it concise and precise and see if anywhere you get between 150 to 200 pages, which covers the entire thing. There is a question from Dr. Shahjaman Khan, which says, how to collect research data from different social media platforms. Well, so you can collect research data. Well, practically, I use mostly the PubMed and other resources. If you are going for media platforms for collection, there might be options of downloading it or requesting them to give access to it. Then in most of the cases nowadays, scientific community gives access, at least if not completely, to a part of it, which will be helpful for you. There's a question from Ashok Tawar which says, what is exploratory sequential design? Explore, yeah. That is nothing but what do you call that? It's explorative research in the sense meaning we are working out on the whole research path as such, and then we are trying to do various aspects which are associated with it. So we are exploring the whole thing. That's why it has been put forth as such. Okay, thanks so much for all the participants. If in case you have got any further questions or any other queries, you can contact Tejasvi and we will be able to help you out.